oranges have the highest amount.
Are you sure? But what if another fruit juice actually contains more vitamin C than ours?
Good question. But how can we find out in chemistry?
Good day everyone. Today we will present our experiment entitled Vitamin C Test in Fruit Juice by Titration. The objectives of this experiment is to determine and compare the vitamin C content in different fruit juice samples using titration analysis.
Okay, first we start with introduction. Vitamin C is known as ascorbic acid. It's a water-soluble vitamin found in citrus and other fruits, berries, and vegetables. It is essential for the repairs of tissues as a collagen. Vitamin C is also important for immune system function and acts as antioxidant. Fruits such as guava, kiwi, papaya, and citrus fruits are among the richest natural sources of vitamin C, essential for immune support, collagen synthesis, and antioxidant protection. Natural vitamin C can be found in fruit like orange, kiwi fruit, guava, and vegetables such as bell pepper and broccoli. It comes with other phytonutrients, flavonoids, and bioflavonoids which may enhance its antioxidant effects and absorption. Synthetic vitamin C is typically made through industrial processes using glucose, fermentations, or chemical methods. It is also available in tablets, capsules, powder, and protect foods without the additional plant compounds. Here are examples of fruits: orange, guava, and lemons. Okay, so what is synthetic vitamin C? Alright, synthetic vitamin C, also known as ascorbic acid, is a water-soluble compound essential for human health, supporting collagen production, antioxidant defense, and immune function. Humans cannot produce vitamin C naturally, so it must be obtained from diet or supplements. Chemical synthetic vitamin C has the same molecular formula as the vitamin C found in fruits, vegetables, meaning that the body recognizes and uses it in the same way. So, how synthetic vitamin C is made? From industrial production of synthetic vitamin C usually start with D-glucose, often derived from cornstarch. The traditional rich stand process involves multiple steps: hydrogenation of glucose to D-sorbitol, microbiooxidation to L-sorbose, the further chemical reaction to produce L-ascorbic acid. While synthetic vitamin C is chemically identical to natural vitamin C, natural sources contain additional compounds such as bioflavonoids, lutein, and other phytonutrients that may be enhanced antioxidant activity and absorption.
And now, let's move on to the section 2, theory. In this section, we are going to dive into the core chemical and scientific principles that makes our experiment possible. For our theory part, I will explain about volumetric analysis. What is volumetric analysis? Volumetric analysis is a method used in chemistry to find the concentration of a solution. This method is also known as titration. It is commonly used in the lab because it is simple, fast, and also accurate. In this experiment, we use volumetric analysis to find the amount of vitamin C or ascorbic acid in the fruit juice. The fruit juice acts as our sample with an unknown concentration. It will react with a solution of a known concentration, which is our titrant. This will help us calculate exactly how much vitamin C is present. Our chosen method is a direct titration. In a direct titration, vitamin C reacts directly with the titrant. This means there are no extra steps required, like in a back titration, making the process much faster and easier to do in the lab. The main principle for our topic is that vitamin C acts as a reducing agent. It reacts with chemicals such as DCPIP or an iodine solution. During the reaction, vitamin C will be used all up, so the endpoint can be seen through a color changes. For example, DCPIP changes from blue to colorless. So this shows that the vitamin C has reacted fully to the experiment. So why do we choose this method for our experiment? We choose this method because it is easy and quick, also gives very accurate results. This method is also suitable for liquid samples like fruit juice that is also used in food chemistry testing.
Hi, now I will explain the methodology used in this experiment to determine the vitamin C contained in orange, guava and lemon using the titration. So, the first one is collection and preparation of sample. First, we collected fresh orange, guava and lemon from a local market. The fruit were washed thoroughly to remove any dirt or impurities. Next, the juice was extracted from each fruit and filtered to remove seeds and pulp. Finally, each juice sample was labeled according to its fruit type to avoid confusion during the experiment. Preparation of reaction and iodine titration procedure. Next, we prepared the reagents for the experiment. A standardized iodine solution was used as the titrant, while starch solution was prepared as the indicator. Before starting the experiment, all glassware was cleaned and rinsed to prevent contamination. For the titration procedure, 10 milliliters of each fruit juice sample was transferred into a conical flask. A few drops of starch indicator were added, and the sample was titrated with the iodine solution until a permanent blue-black color appeared. The volume of iodine used, also known as the titrate volume, was then recorded. Repetition of experiment and data analysis. To improve the accuracy and reliability of the results, each fruit juice sample was titrated 3 times under the same conditions. The titer values obtained were recorded and averaged. The average titer value was then used to calculate the vitamin C concentration in each fruit sample. Finally, the vitamin C contents of orange, guava and lemon were compared to determine which fruit contained the highest concentration.
Next, we will discuss the result obtained from the iodine titration. The table shows the titrate value for the 3 fruit juice sample task. Guava juice recorded the highest titrate value of 30 ml, followed by orange juice, which is 13.80 ml, while lemon juice recorded the lowest titrate value of 10.0 ml. The titrate value represents the volume of iodine solution required to react completely with the vitamin C present in the sample. A higher titrate value indicates a higher amount of vitamin C because more iodine is needed to reach the endpoint of the reaction. Therefore, based on the titrate value obtained, the guava juice is expected to contain the highest amount of vitamin C among the fruit sample task. Based on the titrate value obtained from the titration, the concentration of ascorbic acid or vitamin C was calculated for each fruit sample. As shown in the table, guava contained the highest concentration of vitamin C at 307.38 mg per 100 grams of fruit. Orange contained 142.07 mg per 100 grams, while lemon contained 103.14 mg per 100 grams. The result indicates the guava is the richest of vitamin C among the fruit test. This is because it required the largest volume of iodine solution during titration, showing that it contained more ascorbic acid. Overall, the result demonstrates the different fruit contain different concentration of vitamin C, which is guava providing a higher level compared to orange and lemon.
In conclusion, the titration experiment was successfully carried out to determine the concentration of vitamin in different fruit juice. Vitamin C reacts with the iodine solution through a redox reaction, and the endpoint was identified by the formation of a permanent blue-black color when starch an indicator was added. The result allows us to compare the vitamin C content of different fruit juice and show that fruits are important natural sources of vitamin C. As an essential nutrient, vitamin C plays an important role in supporting the immune system, collagen synthesis, and tissue repair. Overall, this experiment enhanced our understanding of titration techniques, redox reaction, and analytical chemistry concepts while demonstrating the significance of vitamin C in maintaining good health.